Good evening, everyone, and welcome to today's webinar, the IVF Laboratory Optimizing Patient Success. Before we get started, I would like to go over a few quick items about today's event. First, all physicians in attendance today will receive an AMA PRA Category 1 credit, and all other providers will receive a certificate of completion for 1 contact hour credit. Additionally, we encourage you to submit questions to Dr. Sakkas using the questions section of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the question and answer session at the end of today's presentation. Now, without further ado, I would like to introduce today's speaker. Dr. Denny Sakkas is the Chief Scientific Officer at Boston IVF, where he actively cultivates new technologies that improve embryo development in our lab. Dr. Denny Sakkas received his undergraduate training at the University of Melbourne, Australia, and received his doctorate of philosophy at Monash University, Melbourne, Australia. Dr. Sakkas earned his PhD at one of the leading reproductive research units in the world. He has extensive experience in clinical IVF and reproductive research. He is internationally recognized, having organized numerous conferences and workshops on reproduction-related topics, and is frequently invited as a speaker at major conferences worldwide. He has published more than 200 peer-reviewed manuscripts and chapters in the field of fertilization, early embryo development, and male infertility. All right, let's get started. I'm going to pass it over to Dr. Sakkas.
Okay, thank you, Alyssa. So today I'm going to talk more about the IVF laboratory and how that can actually act to optimize patient success and improve patient success. So as we all know, the patient or the couple's journey is a very complicated one. It starts with the consultation and counseling and their acceptance of having to do infertility treatment. They then enter into this sort of roundabout or roller coaster ride of treatment, including stimulation, and then they enter into the lab process, which includes egg collection, sperm preparation, sperm analysis for the partner, maybe egg vitrification, or technologies that have been around for quite a while now, but have made a massive impact in IVF. Things like intracytoplasmic sperm injection, which I'll talk about a bit later. Of course, growing their embryos in vitro, and then maybe freezing their embryos or some sort of analysis of their embryos. And of course, all that leads to some type of embryo transfer where their embryo is put back into the uterus. They then unfortunately undergo more drugs, they undergo a pregnancy test, and then the big sort of roller coaster ride strikes of success or failure. And if they fail, they go through all this process again. So I'd like to sort of talk today about how maybe the laboratory can impact on that success or failure and how it impacts the couple's journey. So this is a diagram I like to show in many lectures, but it's applicable to patients and so their understanding of how reproduction failure or success may actually be impacted by the different parts of the couple. So the egg is the queen of reproduction. It probably impacts the greatest on actually success or failure, and you could roughly average that to about 70 or 80% of the importance is related to the egg or the egg quality. The sperm obviously does something, so the sperm actually has an impact, but it's probably much less than the egg, I would say. Once the sperm and the egg get together and you have an embryo, obviously that is key to actually the way the successful outcome of an IVF cycle. And then once that embryo is put back into the uterus, obviously the uterus actually has a strong role in actually whether that embryo will implant and then go on to a pregnancy. So all these are key factors in the success or failure of a reproduction cycle. And they're things that we have to, in the laboratory, treat with kid gloves, if you want, when we actually access them in the lab. So one of the things we're very interested in is how technologies can impact this patient journey and how we can improve the collection and the treatment of these eggs and embryos. And there's a number of things that we're examining, and I won't go into all of these, but I'll talk briefly about some of these and also talk a bit about how we do things in the laboratory. So the first thing everyone hears about is the Internet of Things, or how the web or the network of IVF, how the network will impact IVF. So the Internet of Things is a network comprised of physical objects capable of gathering and sharing electronic information. And that we see every day. You know, everyone that has an Amazon Echo or something in their house is part of the Internet of Things. But these things can also impact the way a patient is treated also. One of the other things I'll talk about is something called artificial intelligence or machine learning, where we're now using the high order mathematics, if you want, to treat the data we get from all our patients and from the treatment that they undergo. I'll talk briefly about another technology that we've got a lot of interest in called microfluidics, and it's exactly that, what it says. It's micro or very small movements of fluid, and how we can use very small movements of fluid to move, in particular in the IVF lab, to move cells around and to possibly help that type of microfluidics treatment to select cells or actually improve the way we grow embryos. So I'll talk about that a little bit. And then I'll talk about a little bit about novel analytical technologies, or the way we analyze embryos in particular, or even sperm. So how all these types of technologies may impact an IVF cycle and the patient's journey. So first I'll talk a little bit about the male, so a little bit about how we may do sperm analysis and sperm preparation, and some of the techniques we use now, like intracytoplasmic sperm injection, and how they may change in the future. So the first thing you have to realize is in IVF, we're trying to do things like in vivo, but it's very difficult to do an in vivo type phenomenon when you're actually faced with the different scenarios we get of patients. So if you look at the natural in vivo progression, you obviously have the oocytes developing in the ovary, the sperm developing in the testes, and then the sperm and the egg get together in the oviduct or the fallopian tube. They grow in the uterus, they implant in the uterus, and then you have a successful newborn. So that's the sort of natural process in vivo. But the different types of IVF treatment all skip part of these steps. So even in insemination is skipping part of this step of actually putting the sperm past the cervical canal, past some of the things that it will normally face, and into the uterus. Obviously, in doing an in vitro fertilization cycle, we're changing a lot of things. We're changing the whole laboratory, or the whole events that occur in the fallopian tube and the uterus. In ICSI, we're changing other events. We're even changing the event of fertilization. And there are other techniques we use where we actually collect sperm from the testes, where we're actually changing the technology of or the process of sperm actually growing right through maturation. So all these processes involve the laboratory in some way, and an intervention by the laboratory. So, and many of these things involve actually taking an embryo or an egg or a sperm outside of the body and trying to deal with it outside in a less natural environment, if you want. So what about some of the technologies we have that could help the patient? So one of the technologies that is interesting is actually, and is now probably becoming more apparent with COVID, is patients actually don't want to come into work or to the clinic as much. So we're looking at technologies that actually offer analysis of the gametes, and in particular sperm, to be done at home. So the patients don't have to come in. So these are type of smartphone technologies where there is an app on the phone, and the patient can actually do the semen analysis at home, download it on an app. There's a little camera on the app. It takes an analysis of the sperm sample, and this can all be done at home. And actually that is then conveyed to the laboratory, where the clinician can make a decision about whether that sperm will need a particular type of in vitro fertilization treatment. So these are some of the technologies that people are looking at. Other technologies or logistic offerings are a bit simpler. So this is another type of remote testing of sperm, where companies offer actually the logistics of actually sending a pack to the house of the man. They can then produce the sample at home, they package it up, and actually the logistics are all organized to actually send that sample to the clinic, and actually have that sample analysed at a clinic without the male actually having to come into the clinic. So these are types of logistic technologies that are being offered now that will ease that patient journey, if you want. And finally, there are other techniques that we're using in the lab to try to actually take that 10 to 15% that's important of the sperm and maximize that. So we're trying to actually use technologies to pick the best sperm. And this is where these types of microfluidics come into practice. So there are technologies now that actually allow you to actually put the sperm into like a maze, if you want, of fluid or an obstacle course, if you want. And it actually mimics a little bit of what happens in the female reproductive tract, where the sperm actually goes through this obstacle course, if you want, and the best sperm actually get to the end. So that allows us to actually then pick that sperm to actually use it to fertilize the egg. There are techniques like microfluidics available, but there are other techniques such as this technique where you can choose a sperm according to its maturation, or that it's made sure that it's developed correctly. And this is a technology where you can actually put the sperm in a dish. The dish actually has a glycoprotein on it called hyaluronan that normally is around the cells that surround the eggs. And what happens is the good sperm will actually bind to this substance. So if you can see this video here, and I hope this works, you can see a lot of sperm moving, but you can see some sperm moving around and then just stopping and actually binding to the substance. These are actually good sperm. And what you can then do is actually come down with a little pipette, and you can pluck that sperm off and actually use that sperm as a type of selected sperm. So it mimics the way the egg would have selected the best sperm, if you want. So these are technologies now being used in many laboratories, including our own, to try to help and pick the best gametes, if you want. So what happens once we collect the eggs from the patient? So these are cumulus cells, and these are the types of cells that have these proteins on the sperm come binding to, and the sperm actually burrows through here normally and fertilizes the egg, which is in the center. So what we normally do is we collect a lot of these eggs from patients by exaggerating their hormones, if you want, by stimulating them. And then once we collect those eggs, we are able to inseminate them with sperm. And one of the techniques we use in the laboratory is a technique called intracytoplasmic sperm injection. And that technique involves catching a sperm with a small pipette and then injecting it into the egg. And I'll just show you a quick video of this technology. So this is a technology that was found in around 1992. The first pregnancy was reported, and you can see here the sperm is actually captured in a pipette. Let me just get this video going, and you can see then the pipette is taken over, and that sperm is then injected directly into the egg. So it's a very powerful technique. It revolutionized the way we treat male factor infertility. So we can virtually treat males with even one sperm if you want, although that's an exaggerated case. But you can see here now the sperm is actually in the pipette here. This is the egg. This is like a shell or the zona pellucida around the egg. The sperm is injected into the egg. It's then aspirated just to make sure that you've broken the membrane, and you'll see now the sperm will actually be injected into the egg, and hopefully that egg will fertilize. So you'll see the sperm coming down now, just there, and you can see the sperm's placed into the egg. And this technique actually revolutionized the way we do the way we treat infertile males. So we're able to treat virtually any male that we can recover sperm from, and that means even recovering sperm from the testes if need be. And it's a very successful technique. Once we do that technique, we normally wait around. So if we did that this afternoon, for example, the following morning, so tomorrow morning, about 18 hours after, we would then examine that egg to look for fertilization. And the way we look for fertilization is we look for these two pools, or these two pronuclei, in the egg, in the developing egg, and that way we know that the egg is fertilized. So these are techniques we use routinely, but we are looking at different techniques to hopefully in the future improve the way we do ICSI. So this is a technology based on microfluidics where sperm actually swim through these little canals that you can see here, and there's the little holes. There are little holes in the canals that are different sizes, and actually some good sperm will actually be trapped by a certain sizes of holes. So what will hopefully happen in the future is we'll be able to select also sperm before we actually inject them. So it will help this capturing process of sperm to actually be able to perform this ICSI technique much easier, and to know that you've actually got a very good sperm that you're actually injecting into the egg. So these are techniques that hopefully in the future we'll be using in the near future, I would hope, because they're already being used in some animal models. What about other parts? So I've spoken a little bit about the sperm, a little bit about how we fertilize embryos, but how do we grow embryos now? And what are some of the techniques we're using to actually look at embryos in the laboratory? This is probably the most simple but also intriguing technology, and it's actually called time-lapse imaging. So this is an embryo, as I showed before, with the two little pools here, the two pronuclei, and this is the development of an embryo over the 5 days. So you can see the embryo going at 1 cell stage. This is the timing here, 30 hours after fertilization. You can see the embryo developing to the 2 cell stage here, the 4 cell stage at about 45 hours. And you will soon see this embryo cleave again to the 8 cell stage. So you can see here it's gone to the 8 cell stage. It will then start to form like a ball, so you cannot count the cells anymore. And that is the compacted stage, or the morular stage. So you'll see these cells now start cleaving again to around the 16 cell stage, but then they actually start forming a ball-like structure. So you can see that ball-like structure there. And then finally, once this is around the 4th or 5th day of development, in the 5th day of development, you'll see a little fluid sac developing in the embryo, and then the embryo will become what's called a blastocyst. So it will actually form 2 parts. So you'll see the sac forming there, and then you'll see the embryo, forming what's called a blastocyst. Let me just go back to the final part again. So you can see here, this is a blastocyst, and the blastocyst consists of 2 parts. It's called the inner cell mass, which actually leads to the fetus, and the trophectoderm, which actually leads to the placenta. So this happens at around the 5th or 6th day of development. But this type of technology, where we can take a video of the embryo in a closed environment, allows us to actually create a lot of data on how we actually examine embryos. Because normally the way we examine embryos is we actually look at them down a microscope, or only at one time. We don't look at them over all the time frame. But these types of video imaging systems allow us to get a lot of data on the way an embryo develops. But normally in the lab, the way we look at embryos, and especially at blastocysts, we actually we look at embryos and actually assess them according to what's called their inner cell mass. So you can see here this ball of cells here that's form around in the center, or the periphery of the embryo. And as I said before, these inner cell mass cells become the fetus, or the actual baby. The cells around the edges are called the trophectoderm. So this is like a sphere, a ball-like structure with a fluid inside it, with some cells to sort of one side, which are the inner cell mass, and the trophectoderm cells that are surrounding the embryo at the blastocyst stage, which go on to form a trophectoderm, or the placenta. So how do we actually know which embryo is a good one? And it's probably the biggest challenge we have in the laboratory. Which embryo out of the 4 or 5 a patient may have will give you the baby, or will not give you the baby? So how do we know that? And it's probably the biggest challenge we have in the IVF clinic or the IVF lab. And you can see how challenging this is by looking at how out of 100,000 patients, how easy it is for us to actually get them stimulated, to get eggs from them. We're very good at getting egg retrievals. We're very good at getting embryos to put back to them. But the actual biggest drop in our ability to actually perform IVF is actually this step here. So we can put embryos back into patients, but most of the time, or a lot of time, the biggest challenge we have is actually picking which embryo will provide a live birth, which embryo that we transfer will give us a pregnancy. We have got much better at that, but this is probably the biggest challenge we have in actually in trying to develop technologies to actually pick which embryo is the best one. And we have a number of techniques, and probably the best technique we have at this stage is a technique called pre-implantation genetic testing. And this involves actually taking a blastocyst like we have here, and from part of the trophectoderm is actually taking a small biopsy, removing some of the cells and actually testing them genetically to see how the chromosomes are actually working in that part of the trophectoderm. So you can see this technique. It looks pretty aggressive if you want. So what we do is we suck up a few of the cells from the trophectoderm, and then with a laser, you can see this small red dot, I hope, we cut down and we actually cut off the cells here, and you'll see these being zapped by the laser, and then you've got the removal. The blastocyst remains intact, and these cells here are then sent over for analysis for genetic testing. So this is a quite invasive technique. It involves the laboratory and more highly trained technicians. And what it tells us is that it gives us a chromosomal aneuploidy analysis of the embryos. And what we do know, if you look at this graph here, this is the age of the patient. This is the percent of abnormal embryos, so aneuploid embryos. And you can see here in young patients, so under 38, you can see on average, if you look here, about 30% of their embryos or their blastocysts that we biopsy will be abnormal, which means a high percentage will be normal. If you go to around 38 to 40 though, you can see this percentage starts increasing dramatically. So we know that the level of chromosomal aneuploidies, or the abnormal embryos in a patient, increases dramatically once a patient is over 38 years of age. And these are embryos that we've done, and you can see on average a younger patient below 35 or 38 will have about a 30–35% chance of having an aneuploid or an abnormal embryo. But this dramatically increases from 50 up to 80% in patients over 40. So this technology of biopsying the embryo is very effective in actually allowing us to understand which embryos are normal and abnormal. And how does this technology work? So when we get this biopsy, you can actually run it through a chromosomal analysis, and the technology that's normally used now is called next generation sequencing. And basically it looks at pieces of all the chromosomes of the embryo, or the biopsy from the embryo, and looks to see if they're actually intact or in pairs, if you want. So you can see here when they're paired, you can see that they're actually, you get normally a straight line if it's a female embryo, and obviously a little kink if it's only one X and one Y. So you can see these embryos that give you these patterns are normally normal. But when you start getting deviations from this line, such as in this case underneath here, you start knowing which embryos would not actually be viable and would not be normal. So this technology now is used routinely in the laboratory, but it does involve an actually invasive biopsy. So that technology that I showed you. One of the things that is quite interesting, or interesting a lot of people, is that sometimes if you take a biopsy from an embryo, the majority of times, if you look at the trophectoderm biopsy from the test, and then you take a second biopsy, hopefully you always get the same biopsy. But sometimes you start seeing differences. So for example, in this case down here, this is the trophectoderm biopsy that showed a small abnormality. And if they took another biopsy, it actually showed that this was actually a different abnormality, or it may have been normal. So one of the challenges we have with this biopsy technique is that we're seeing some abnormalities in parts of the trophectoderm which are not necessarily mirrored by the whole embryo. So it's not always a 100% technique, but it is a very, very powerful technique for selecting the best embryo. But what other methods could we use? And this is somewhere where we've been very interested in, that would not really necessitate the use of a biopsy. So we didn't have to actually take cells from the embryo. Are there technologies that we could use that are non-invasive, that we could actually in some way examine the embryo by looking at something else that the embryo is doing in terms of in the way it's growing, or possibly in the way it's actually putting something out or secreting into the culture media? So one of the things we're very lucky in, we grow embryos in these very small droplets of culture media. And these culture media allow us to do two things. So one, we actually can view the embryo. So we're able to visualize the embryo, as I showed you before in the video image. But also these embryos, we know that they produce factors and secrete them into their surrounding. So two things we can do is actually we can analyze embryos by the way they grow and maybe patterns of growth, but also the way they actually metabolize, or they actually factors they produce into the liquid surrounding them. So one of the technologies that I mentioned before was this time lapse, or these video imaging, and what I showed you before were videos taken using this system here. So the embryos are grown in very, very small wells. And basically there's a camera that goes underneath each embryo every 10, 20 minutes and takes a snapshot of how that embryo is growing. But from what I showed you before from the video, you can actually get a lot of data. And all this data, you can get pictures, you can analyze those pictures for certain patterns within each frame of the movie you get from, or the time-lapse image you get. And using artificial intelligence, you can actually analyze the time, the quality of the embryo, patterns within the embryo, and use things like recognition software, similar to how your iPhone now recognizes your face to know that it's you. You can use all that information to actually teach yourself to say which embryo is the one that's going to give me a baby. And this type of time-lapse imaging assessment using artificial intelligence is now becoming very popular and becoming used in many studies. And this is just an example of one study where they use this machine learning or artificial intelligence to predict which embryos would become a baby, basically. And this is a very preliminary data, but basically this study showed that they were able to predict with around 90% accuracy using time-lapse imaging and using artificial intelligence to actually recognize which embryo was most likely to give you a fetal heart pregnancy, so to establish a pregnancy. So many people are working on this type of technology now, and it's looking quite promising in terms of something that hopefully in a year or two will actually be used in laboratories in some fashion. And that might be through video imaging, or actually even through still images. So this type of technology is where you have a great technology such as the time-lapse imaging of embryos, married with very high-level mathematics and artificial learning is very promising in terms of possibly being able to help us pick which embryo is the best one. There's some other techniques though that are interesting, and this is a study that came out quite a few years ago now from Harvard, where they were able to do the biopsy technique that I showed you before, but actually get the same information just by looking at DNA that the embryo was actually secreted into the culture media or its surrounding environment. And this technique actually has now instigated quite a lot of studies, and I'll just show you some data from one of these studies that we participated in. So this technology, instead of invasive PGT, where you actually take a piece of the blastocyst, is called non-invasive PGT. And what we do is actually at around the 4th day of development, very early in the development, we actually take each embryo, we wash it through some culture media, so the media that it's growing in, and then early in that morning, we actually put them into little droplets and let them sit there for about 30 hours, till about the 6th day of development. And what we do then is hopefully that small little embryo will secrete some signal, which in this case is DNA, into the culture media that will allow us to do this technology of next generation sequencing, to do an analysis to know if that embryo is euploid or aneuploid, exactly the same we do for the analysis when we do the biopsy. So these embryos then, we wait till about the 6th day of development. We then take the embryo just as we would for a biopsy, but we freeze the embryo. But then we actually, from the media that it was growing in, we're able to take all that media, put it in a tube, and send that tube for analysis, just as we would if we actually had taken a biopsy. So what happens when we do that? Well, we've just published a study with a multi-center with a lot of other groups, and we wanted to see if you compared the technique we use now, the biopsy technique where we take a biopsy, compared to the media, how often they would agree. So these are examples of, this is a trophectoderm biopsy, and this is the media, and you can see here the next generation sequencing readout was the same. You can see here the trophectoderm was slightly different from the spent media biopsy, and you also get, sometimes you get an abnormal embryo from the trophectoderm, but you also get an abnormal embryo from the blastocyst media, but they're not always exactly the same. So we've done this study now, and in just over 80% of the time, or nearly 80% of the time, and this is in 8 different clinics looking at this technology, around 80% of the time, the media, so what you find in your culture media, agrees with the trophectoderm biopsy. And this is, I think, a very promising technique, which will hopefully allow us to move away from this invasive technology of biopsy to a less invasive process. And I just remind you that 80% is not too bad, because even when we do a biopsy twice from an embryo, it's not 100%. It's probably around 90–95% that they actually agree with each other all the time. So I think this is a very promising technique, hopefully in the future, that may be adopted by clinics. There's more to just the genetics of an embryo, though. So, you know, there are other ways of looking to see if an embryo is healthy or not healthy. And I like to show this stage because sometimes with the genetics or the growth, the morphology, we don't always see everything, all the information that we need. And this is just an example of picking 3 different people: Winston Churchill, Roger Bannister, and Jimmy Fix. Obviously, Roger Bannister and Jimmy Fix were very fit. They were, you know, they were people that were well known for exercising. Winston Churchill on the left was overweight. He smoked cigars and he drank. And so, but it's interesting because if you actually looked at them morphologically, you may not understand who lived the longest. And I'm sure you know this is a loaded question. So if you look at who lived the longest, obviously Winston Churchill lived the longest. The quality of life may not have been that great, but it is interesting that sometimes just by looking and examining something at a more superficial level, you may not get all the information. So one of the things that we're interested in looking at embryos is actually probably looking more at metabolism of embryos. So, and trying to understand which embryo is the fittest and which is acting the best. And we've been working in the past few years of looking at a non-invasive technique for actually measuring metabolism. And these, if some of you remember some of your biochemistry, these 2 markers, NADH and FAD, examining mitochondrial function. So basically, they're telling us which embryo has the best metabolism. And what we have here, and I'm just going to show you some videos of some embryos that we've tested by by looking at their metabolism, but this is a technology which actually doesn't harm the embryo. We don't have to color it in any way or add anything to it. This is their natural fluorescence, if you like, which allows us to understand which embryo is good and which embryo is bad. And I'll just show you a quick video here. These are mouse embryos, and here you can see these two different metabolic activities tell us which embryos are acting in different ways. And you can see some embryos that are blastocysts here that are in a different mode of metabolism compared to other embryos in their same cohort here. So we believe this type of technology will actually also be very helpful in allowing us to understand which embryo is the best one when we're transferring them. We've actually done this technology now with human embryos, and these are blastocysts, and you can see here we can actually scan a blastocyst. We can look at the inner cell mass here and the trophectoderm. We can get a lot of information about the metabolism of those embryos, and we're getting a lot of information now that we're examining. It appears we can tell which embryos are growing faster, which ones are at the 5th day of development, which are the 6th day of development. We can see differences in their inner cell mass and the trophectoderm, and we have some preliminary evidence that there's a possibility that we could tell which ones are chromosomally normal and ones that are abnormal too. So we believe this might be a powerful technique, possibly not by itself, but possibly as an adjunct also to other technologies that may be developed in the future. So finally, I think one of the biggest challenges we have is actually picking the embryo, the best embryo. But I think we have a lot of technologies now that are non-invasive that we'll be able to use to actually look to see which of these blastocysts from a patient, when they have 3 or 4, which one would be the best one to put back. And this will not only help them get pregnant, but most likely it will help them get pregnant quicker, because we'll be able to pick the best embryo earlier and give them the best chance of actually of completing that patient journey without having to go through the roller coaster and the loop of actually repeating it several times. And I think that may be just as important for the patient, so they don't give up on trying IVF, and actually can hopefully succeed much earlier. So finally, I think some of these technologies will be very promising. There are other technologies that we're examining that hopefully might even help even further in the future. So finally, how will a lab maybe look in the future? And this is not now or not in the next few years, but possibly in the future, we will have little sort of chips, if you want, microfluidic chips, where we can grow embryos in little wells and examine them in real time using various technologies, hopefully non-invasive. So you could imagine each of our embryos would be grown in these little wells. We could feed it with different culture media, and we can actually test that culture media at the same time. So we could actually, using microfluidics, you can run these media or the samples, and you could test them for maybe DNA, you could test them for metabolism markers, you could visualize the embryos, and all this may be in a small little incubator, a small unit that we would be able to grow and test the embryos at the same time, and possibly also freeze them and maintain them for future use. So I think there are even though we're very good at doing things that we do in the laboratory, and we have a lot of data, and our pregnancy rates have improved, I think in the future we will further improve things, and using novel technologies, I think will help that patient journey, and the patients, actually the couples, achieve their goals much quicker and with greater success. So thank you. If you have any questions in the future, I'm happy to answer them. You can always contact me at my email address at Boston IVF. So thank you for listening.
Thank you, Dr. Sakkas. We are now going to begin answering questions submitted during today's presentation. As a reminder, you can still submit questions through the questions pane in your attendee control panel. All right, so our first question is, what do you think will be the next new technology in the laboratory?
I think some of the ones that are probably looking more likely are something like the non-invasive culture media testing. There are some companies that are looking at that already. As I said, we're just part of a large multi-center trial using that technology. So I think that's probably something that is most likely going to be a technology that people will use in the very near future.
Great, thank you. The next question is, does the biopsy decrease the viability of the embryo?
So it's an interesting question, because one of the things we're doing when we're biopsying an embryo is we're testing whether it's chromosomally normal or abnormal. And we know that when we transfer a chromosomally normal embryo, the pregnancy rates are very high. You know, they're well over 50%, even 60% live birth rates. But possibly, if we don't know if actually the biopsy has harmed that embryo, would that pregnancy rate have been 65? Would it have been 70? So we really don't know that, because we think we're very successful at actually getting that chromosomal information. All indications are, and there are a lot of studies on this, that the biopsy doesn't, at the blastocyst stage at least, doesn't cause any harm. Many years ago, we used to biopsy at an earlier stage, at the 8-cell stage. There are some indications that although it gave us the correct information about aneuploidy or euploidy, it didn't. It may have actually been harmful in a way. But the indications at the moment, if all the studies on the babies born after biopsy of blastocysts, the pregnancy rates indicate that it's not really that harmful.
Great, thank you. I have another question. Is the blastocyst biopsy analogous to the CVS?
So
like,
obviously much earlier.
Yeah.
It's looking for the same information if you want. So you know, you're after the trophectoderm if you want. So you're looking at the same tissue. The technology they use to actually get the analysis is based on next generation sequencing. So in effect, it is very analogous technically. Obviously, in terms of the stage that it's being performed is much later. So if you're looking at a CVS, but in effect, yes, it's an interesting question. I've never thought of it that way, but in many ways, they're very analogous. Even probably your chances of having an abnormal result are probably pretty similar, to be honest.
Great. The next question: can we improve poor embryos in the laboratory?
So I think this is sort of going further down the road. You can definitely harm embryos in the laboratory by growing them in the wrong conditions, maybe not being as careful about temperatures, about the environment you grow them in. But many people are now looking at ways to possibly improve embryos in the laboratory, and that may be by simple techniques like keeping them in the one environment all through the process. So quite a few years ago, we used to grow embryos and we would take them out every day to look at them, because we wanted to know how they were growing, and the patients wanted to know how they were growing. But systems, for example, like the time-lapse systems, allow us to look at the embryo, but we don't remove it from the environment it's in. So it's in a very stable environment. So people believe that actually as simple a thing as actually leaving the embryo in the one static environment for the 4 or 5 days that it's growing is improving the embryo. Other techniques people are looking at is actually improving the media or the fluid that we grow embryos in, and possibly looking at factors that may actually help the embryo grow a bit better. So maybe introducing some growth factors or some factor that would help a poorer embryo actually grow faster or grow better. So they're technologies people are looking at. And then finally, people are looking at microfluidics, where the media is being changed or is mimicking maybe a bit more closely what's happening in the uterus or the fallopian tube. So I think in the future, people hopefully may be looking at techniques that will actually make embryos grow better in vitro. The one thing we don't know is we don't really have that much information on how human embryos grow in vivo. So it's very hard for us to have like a gold standard that we know this is how everything's done in vivo. How do we do it in vitro? So some of the information we are missing is actually what does a human blastocyst really look like when it's grown in vivo? We only have very limited information on that.
And this is currently our last question. Where else could artificial intelligence help in IVF?
So artificial intelligence is likely to help in places that we can't even imagine. So I think in the main part, it will help in the laboratory, I think, as we're already seeing, but it will also start helping us in actually knowing how to treat patients. So actually personalizing their treatment a bit more. So things like a patient that comes in may fill in a questionnaire, and based on that questionnaire, we will have a better idea of how to give them, what type of drugs we should give them, what type of chance they have of achieving a pregnancy. So I think artificial intelligence will change a lot of things in terms of streamlining the process for that journey for the patient and streamlining their journey through IVF and hopefully improving their chances of achieving a pregnancy.
We do have one more question. Any experiments with CRISPR in the embryo stage to prevent or cure disease?
Are there experiments? There's obviously research being done in this field. There's already been quite a 2 or 3 publications looking at trying to use CRISPR or gene editing to improve embryo or to change the genetics of an embryo, and this could prove a powerful tool in patients who have single gene mutations. So a patient, for example, that is a carrier for cystic fibrosis, for example. So it could prove a powerful tool in improving their chances of having a normal embryo, whereas now we would screen those embryos and we may have a 1 in 4 chance or a 1 in 8 chance of getting the right embryo. CRISPR or gene editing may be able to actually improve the chances of having more embryos that are normal by having the ability to change them. This technology, though, is very far away, I think, in terms of clinical application, and so I think that's probably it's an interesting research topic at the moment, but I think for it to be clinically applied, it will have to go through quite a stringent testing, because people are still not sure when you do the gene editing whether it actually is successful all the time. So I think even though that's a very powerful technique, I think applying it to embryos is still a way away.
Thank you, Dr. Sakkas, and thank you everyone for attending today's webinar, the IVF Laboratory Optimizing Patient Success. If you have any other questions, please contact myself, Alyssa Cooper, at ecooper@bostonivf.com, or my cell phone is 978-302-2958. Once you leave today's webinar, you will receive a survey on the presentation, and we would really appreciate it if you would complete the survey and provide your feedback to help with future educational webinars. You will also receive a follow-up email within 24 to 48 hours with a link to view a recording of today's webinar. On behalf of Boston IVF and Dr. Sakkas, thank you for joining us tonight, and have a great rest of your night. Thank you, Dr. Sakkas.
Thank you. Bye.
Thank you, everyone. Bye.